Hello everyone, welcome to 8th day of genetics. Today we are going to discuss one of the receptor protein defects, that is familial hypercholesterolemia. Familial hypercholesterolemia is caused by the mutation in the gene which encodes for LDL receptor. Because of the mutation in the LDL receptor, there is inadequate removal of plasma LDL by the liver. Hereafter, I am going to tell some of the important facts you have to memorize. So, the most common, that is around 80 to 85% of the mutation is in the LDL receptor gene. Fine? Yes. And actually, these genes will code for apolipoprotein, that is ApoB100, the ligand for LDL receptor, then proprotein convertase subtilisin kexin type 9, okay, that is better abbreviated as PCSK9. Actually, this reduces the expression of LDL receptors by downregulating their recycling and degrading them in the lysosomes. Fine. So, these are the two important changes which happens as a result of this LDL receptor mutations. So what happens? There will be increased serum levels of cholesterol, which give rise to premature atherosclerosis and the greatly increased risk of myocardial infarction. Okay, but remember, the familial hypercholesterolemia, which is caused by this LDL receptor mutation, is one of the most frequently occurring Mendelian disorders. That is 1 in 200 individuals. Okay, yes. So, what are the normal clinical manifestations? Okay, clinical features. Remember, skin xanthomas, coronary, cerebral, peripheral vascular atherosclerosis, myocardial infarction. It can occur even before the age of 20 years. Okay, yes. And I have told you premature atherosclerosis. So these are some of the clinical manifestations of familial hypercholesterolemia. But before going into this familial hypercholesterolemia, you should understand what is the normal cholesterol metabolism and transport. You all know that the cholesterol is derived from the diet as well as from the endogenous synthesis, and this dietary triglycerides and cholesterol, it will be incorporated into chylomicrons in the intestinal mucosa. Fine. And these chylomicrons will be hydrolyzed by endothelial lipoprotein lipase, LPL we call it as, in muscles and fat. And the chylomicron remnants are rich in cholesterol. They are finally delivered to that of the liver. Some of the cholesterol will enter the metabolic pool, and some other amount of cholesterol is used for the synthesis of bile acids, and the endogenous synthesis of cholesterol and LDL begins in the liver. So the most important pathway what we have to focus here is the LDL synthesis which occurs within the liver. Okay, yes. See, this is the low density lipoprotein metabolism. Okay, what I have told you only they have demonstrated here. Okay, yes. Okay. And remember, the most important thing is B100. ApoB100 is the lipoprotein which is present in LDL. That is the most important point you have to remember here. Okay. Once that is done, see, this is the normal regulation of the cholesterol metabolism with the help of the LDL receptors. See, okay, we have the LDL receptors. The LDL will binds with this LDL receptors, and what is this process called as clathrin? Okay, this is called as clathrin mediated receptor endocytosis. Clathrin mediated receptor endocytosis. Okay, clathrin mediated receptor endocytosis. Okay, and remember, this PCSK9 will inhibit the recycling of this LDL receptors. Okay, fine. Then the rest of the pathway you have already studied in your biochemistry. That is, okay, what is it about the synthesis of these receptors from the Golgi apparatus, endoplasmic reticulum. Okay, yes. So whenever there is an oversupply of cholesterol, what will happen? It will inhibit the synthesis of LDL receptors, and at the same time it will also inhibit the synthesis of cholesterol, and finally the excess cholesterol will be converted into what we call as cholesterol esters. Okay, and this cholesterol will be also diverted for the formation of cell membrane, steroid hormones, and bile acids. Okay, so these are some of the important points you have to remember. Okay. So there are some four important what processes which happens in this metabolism. Number one is that the cholesterol will suppresses the cholesterol synthesis within the cell by inhibiting the enzyme called as HMG-CoA reductase. You know, the HMG-CoA reductase is the rate limiting enzyme in the synthetic pathway. The next is that the cholesterol will activate the enzyme acyl-CoA cholesterol acyl transferase. This is responsible for the esterification of the cholesterol. So though whenever there is an oversupply of the cholesterol, the excess cholesterol will be converted into cholesterol esters. And next is cholesterol will suppress the synthesis of LDL receptors, so it will protect the cells from excessive accumulation of cholesterol, and it also upregulate the expression of PCSK9. I have already told you that the PCSK9 will be reducing the recycling of LDL receptor. It causes the degradation of the LDL receptors. Okay, yes, okay. So these are the important points you have to remember. Okay, that is just the basic points you have to remember, as well as in case of familial hypercholesterolemia. So far I have discussed what are the two important changes, that is what the APL, am I right, ApoB100. Okay, the problem is at ApoB100, as well as in case of PCSK9. You know what is the role of PCSK9, what are the normal clinical manifestation, and what is the normal cholesterol metabolism and transport, and remember these four processes. Okay, now let us see what are the impact of these genes. Okay, as a result of the mutation, what are the problems which is going to happen? See, whenever there is mutations in the LDL receptor gene, okay, what will happen? The catabolism of the LDL by receptor dependent pathway will be affected. So the plasma level of LDL will be increased. So that is the most important thing. Okay, the most important thing. Okay, yes. So next, another one, whenever there is a mutation in the genes encoding ApoB, what will happen? Yes, it will, the mutant ApoB, it will reduces the binding of LDL molecules with the LDL receptor. So whenever there is a decreased binding of LDL with LDL receptor, automatically the level of LDL in the serum will be increasing. And activating mutation in the PCSK9, what will happen? It will reduces the number of LDL receptor by increasing their degradation, and it will prevent the recycling. Okay, yes. Okay. So that's the most important point you have to remember. Then we have different types of what mutations. Okay, around we have 5 to 6 types of mutations are there. Okay, yes, let us study all those mutations with the help of this simple picture. So in class 1 there is a defect. The defect is at the synthesis. In class 2 transport. In class 3 binding. Class 4 clustering, and class 5 it is recycling. And there is another class, that is the class 6, which is because of failure of the targeting of the LDL receptor to the basolateral membrane. Okay, the LDL receptors cannot be targeted against the basolateral membrane. So these are the 6 classes. There is no need that you have to remember in what in detail about in what in depth about what are the problems in each of these 6 variants. But just remember 1, 2, 3, 4, 5. 1 synthesis, 2 is transport, 3 is binding, 4 is clustering, and 5 is recycling, and 6 the problem is what? The failure of initial targeting of the LDL receptor to the basolateral membrane. Okay, these are the important points you have to remember. Just remember this picture. This is just familial hypercholesterolemia. It won't be asked as a short note question. This is just for solving what multiple choice questions for your exam. Okay, just remember these very basic points: synthesis, transport, binding, clustering, recycling. Finally, in class 6 mutations, it will result in failure of initial targeting of the LDL receptor to the basolateral membrane. So the role of LDL receptor in the cholesterol homeostasis has been well understood with this. Okay, am I right? With this, we understood how far the LDL receptor is very important in maintaining the cholesterol homeostasis. So lot of drugs which are targeted against each of these proteins has been tried for what hypercholesterolemia conditions. Okay, fine. Yes. So these are the important point you have to remember. So xanthomas, premature atherosclerosis, myocardial infarction before the age of 20 years. All these are the characteristic features you have to remember. Okay. So hope you have understood some of the basic points about familial hypercholesterolemia. Thank you.